Hello everyone, we are presenting two-photon excitation microscopy for the study of living cell and tissue by Penninger and Piston in 2013. So today we are exploring the world of fluorescent microscopy, focusing on two-photon excitation microscopy as a viable alternative to confocal fluorescent microscopy. So we will touch on basic principle, historical context, and then delve into comparative analysis, covering principal advantages, limitation, practical consideration, and also example use cases. First, we have a wide field, bright field microscopy, which dated back in 17th century. So how does it work? Is that it uses lamp or any similar light source to produce broad illumination, and this light will pass through a condenser lens here, which focuses it onto a sample here. And then the light will interact with the specimen in various ways, depends on the sample properties, and the objective light here will collect the light on specimen and focus it to form an image, what we see here. And now what is it used for? It has various purposes. First is that it's widely used to examining a variety of biological sample, particularly those with thickness less than 10 micrometers. So this make it suitable for studying specific protein organelle within cells such as mitochondria. And it also a workhorse in investigation of intricate structure across various fields of science. And in field of histology, it played a critical role. It is extensively used to examine cellular structure and detect abnormalities in medical samples such as biopsies for cancer diagnosis. And what are the advantages of it? So first is that it is well suited for live cell imaging, allowing researchers to observe dynamic processes in real lifetime. This makes it ideal to capture dynamic cellular events such as cell division, cell migration, and so forth. And it also achieves high sensitivity, making it effective for detecting low abundance molecule within cell or tissue. However, there are limitations. First is that it's only effective for thin section, which is something that less than 10 micrometers. And it also provides 2D images, which limited in discerning structure and depth information. So in summary, wide field microscopy structure are imposed in 2D images. Therefore, they are limited in discerning structure and depth information, especially in thicker sample. So what have been developed to address this limitation? So the researcher have employed various strategies to improve the traditional microscopy, such as they stain a specific neuron subset using calcium-sensitive dye to highlight cellular activity. So additionally, they meticulously section specimen into thin slices to obtain clearer images. So despite this advancement, there remain a risk of inadvertently severe delicate structure such as dendrite. And another major improvement came with advancement in staining technique and imaging, which directly contributed to development of fluorescent wide field microscopy. So this innovative approach has revolutionized our capability to visualize cellular processes and molecular interaction in real time. So in summary, the development of fluorescent wide field microscopy have significantly improved limitations such as structure and limited depth information. As I mentioned before, the development of fluorescent wide field microscopy is considered a major improvement in microscope world. However, let's go back to basic and understand what is light and fluorescent. So what is light? So light is electromagnetic radiation composed of photons, each carrying energy proportional to its frequency. And as for fluorescence, it is a phenomenon found in certain substances such as dye or fluorophore occur when these substances are exposed to light. Typically will be UV light or ultraviolet light. So upon absorption, they undergo excitation, so transitioning to higher energy level. What happens is that from ground state, it will jump to excited state. And following the excitation, the electron typically doesn't return directly to the ground state, you see. Instead, a stepwise descent occurs, so emitting photon in varying frequency. So the emitted photon carries significant energy, falling within ultraviolet emission or visible or infrared range. So it actually depends on the size of energy gap between excitation and ground state. As for UV emission, it has a large gap, so the emitted photon will be fall into UV range, which is something that we can't see with our human eyes. And as for the visible light, it have an intermediate energy gap, and yeah, the photon will be fall into a visible spectrum, which is something that we can see. And as for infrared, is that it has a smaller energy gap, and the photon will be fall into infrared range, which again something that we can't see. So in summary, in fluorescence, a specimen excited by a specific wavelength returns to its ground state by emitting light in the color of the wavelength emitted, releasing part of the absorbed energy.
So now that we understand the concept of fluorescence, let's have a look at fluorescent wide field microscopy, which was developed as an alternative to bright field wide field microscopy in the 20th century. So how does it work? Well, at first, fluorescent dyes or proteins are used to label specific components within the specimen. Now you can have a look at the figure on the right, and as you can see here, the excitation light directs light of a specific wavelength onto the specimen from the side. Note that this is a pretty big difference from bright field wide field microscopy, where you're looking through the objective and sample directly at the headlight, as you can see here. The dichroic mirror plays a pretty important role in the next step, as it reflects the excitation light toward the specimen, while also allowing the longer wavelength emission light from the fluorophores to pass through for imaging. Next, the fluorophores then absorb the excitation light and emit it at a longer, less energetic wavelength, which then passes through filters that separate the excitation light from the emission light. And then finally, the emitted light is collected and used to form an image of the specimen. So what is it used for? Well, the sample thickness range increased to about 50 micrometers, allowing for the imaging of specific proteins or organelles within cells. A more specific example in cell biology would be that this method can be used to visualize dynamic cellular processes and protein distributions in real time. So to summarize, we can see that fluorescent microscopy illuminates labeled fluorophores in a specimen from a side angle with specific light wavelengths, capturing emitted light through filters to produce high contrast images. Fluorescent microscopy was a definite improvement, but there's still some limitations. But first, let's talk about its advantages. The first advantage would be specificity and selectivity. So fluorescent microscopy allows for a visualization of specific cellular components within complex environments using fluorescent labels. It is also highly sensitive for detecting low abundance molecules, and it enhances contrast. This goes back to what we talked about earlier. So if the sample absorbs about 1% of the light and reflects it back, it will be 1% versus a 0% dark background. Now compare that to bright field microscopy, where you will have to differentiate between about 100% and 99% because you're looking directly into the headlight. Now let's talk about limitations. Well, first of all, we have an issue with background noise from out of focus light or scattered fluorescence, which reduces the image clarity, especially in thicker samples. We still have limited depth information because it's still technically a 2D image. And then there's photobleaching and toxicity, which can cause damage to the tissue, especially if high-intensity light sources are used for extended periods. Photobleaching involves the light-induced degradation of fluorophores over time, which reduces fluorescence, while phototoxicity refers to the cell damage caused by intense light exposure, ultimately impacting the cell health and experimental outcomes because it puts stress on the cell. If we have a look at the images on the right here, we can see we've pretty much come a long way. On the top left here, we can see an image that was acquired using wide field, bright field microscopy. And on the right side, we have a thin sample, not more than 1 micrometer, of chicken growth cones, which are tips of neurons acquired from a fluorescent microscope, which obviously has much more information concerning structure and depth here. However, in thicker samples, and this can be seen here on the bottoms, the image gets blurred because the fluorescence just scatters too much. And this is an image of a mouse intestine. So in summary, we can see that fluorescent microscopy partly resolves the problem of wide field, bright field microscopy, specifically when it comes to discerning structures and just acquiring some depth information. But there still is only limited depth information, and we have new issues such as photobleaching. Another breakthrough then was the invention of confocal microscopy in 1955. How does it work? Well, now the light source is actually a laser, so a laser beam is focused onto a specific point in the specimen. This in turn allows for point illumination, where only the illuminated point fluoresces, minimizing out of focus light. It is not perfectly just that point, as we will see, but it is definitely more concise than fluorescent wide field microscopy. The scanning mirrors rapidly and precisely deflect the laser across the specimen in a raster pattern, enabling systematic image acquisition. You can see the scanning mirrors over here. A so-called pinhole aperture is then placed in front of the detector to block any excess out-of-focus light. And finally, the optical sections collected from these scans that are collected in this raster patterns, point by point, are compiled to construct a detailed 3D image of the specimen, which is also called optical sectioning. In the image on the far right, so over here, we can see the stacked sections, optical sections, that are then put together to form a 3D image, as you can see here in the bottom. And that is a 3D image of a fruit fly brain, actually. And just to make this maybe a little bit more clear, if you look at this illustration on the right here, you can see that even though we just illuminate a point on the focal plane, and this is marked by the pink light, by the pink lines, we still get some amount of out-of-focus emission light, but the in-focus emission light, which is this thicker pink line here, only that will pass the pinhole and reach the detector, forming a sharper image. So what is it used for? Well, the sample thickness range has further increased to about 100 to 200 micrometers, which allows for imaging of detailed structures in thicker tissues or complex cell assemblies such as organoids or tissue sections. And one more specific example would be 3D images of neurons and brain circuits and detailed studies of neural connections and synaptic structures within brain tissue. So to summarize, we can see that confocal microscopy achieves what's called optical sectioning by using the aforementioned pinhole to block any out-of-focus light, allowing only in-focus light from the focal plane to reach the detector. And next up, let's talk about confocal microscopy. What are its limitations? But before we specifically address the limitations, let's talk about its advantages. So first of all, we have the removal of background noise with the use of the pinhole, which provides clear, detailed images because the scattered light is now excluded mostly, especially in thicker samples. Next, we can now also allow for optical sectioning, which allows for 3D reconstruction by imaging all the successive focal planes and then stacking them together into an image. And then we also again achieved improved contrast compared to wide field, bright field, and fluorescent wide field microscopy because of the use of the pinhole and the laser illuminating just one small point in the sample. It's really nice to see that as well here on the top right, where we've seen this image before that was acquired using a fluorescence wide field of a neuron and dendrites, and then on the right we can see a cell structure using confocal that just has a way higher resolution and more structure and depth information. But there's still some limitations, and I briefly mentioned this earlier, because the light still scatters and there's absorption of light in the out of focus area, specifically in thicker samples, even though we're using a laser that is just supposed to illuminate one specific point. And you can see this in the picture on the bottom right, where the fluorescence is excited not just at the focal plane, but also slightly above and below. More specifically, light from the focal plane can scatter and then be rejected by the pinhole, which would reduce the signal. The excitation light itself may scatter, not even reach the focal plane, which would also reduce signal. And the fluorescence from outside the focal plane may scatter back through the pinhole, increasing noise, especially in thicker samples. Also, we still encounter issues with photobleaching, phototoxicity, and while that might not be fully preventable, it's not good that it also happens outside the focal area itself. So in summary, we can see that confocal microscopy provides 3D reconstruction, allowing clear discerning of structures up to 200 micrometers and at different imaging depths, but we still encounter some issues such as photobleaching and phototoxicity outside the focal area, and signal loss and noise increase, specifically in thicker samples.
So now we're going to dive into the alternative to confocal microscopy, which is two-photon excitation. It was first implemented in 1990, so it's relatively new in the world of microscopy. The theory behind it, however, was introduced already in 1931, but wasn't able to be executed until the development of both lasers and more advanced imaging equipment. So how does it work? First, as the name suggests, TPE uses two near-infrared photons which have longer wavelengths, which means lower frequencies, which in turn means less energy. These two photons are absorbed almost simultaneously by a fluorophore, causing it to become excited as we see in the top right image. So as we've seen in the introduction, S0 is the ground state and S1 is the excited state. But now for TPE we have a new intermediate state that these dotted lines refer to. It's actually not a state per se, but just a really small window of time between the first and the second absorption of energy. Due to this very short-lived intermediate level, achieving excitation in TPE requires a high photon density. What this actually means is that for the fluorophore to reach the energy threshold to achieve the excited state, the absorption time between the first and second photon must not exceed this brief period, which is in order of femtoseconds. Otherwise, it would return to its ground state without being excited. So it starts in the ground state, goes through the intermediate level by the first absorption, and then almost instantly absorbs the energy of the second photon to reach the excited state. This is accomplished using pulsed lasers that emit light in intense short bursts. This ensures that the absorption of both photons occurs before the intermediate state decays. And by high intensity, we don't mean that the photons themselves carry more energy, but rather the flux of light is more densely packed with photons. One of the other main features of TPE is its nonlinearity. In one photon excitation, reducing the laser power by half would also cut the fluorescence in half, because it scales linearly with the photon concentration. In contrast, two-photon excitation is nonlinear, scaling with the square of the photon flux, which is represented with I squared, because the fluorophore has to absorb the energy from two photons at the same time, which has a much lower probability to happen. Which means in TPE, cutting the laser power in half reduces the fluorescence to a quarter. As we've already discussed in confocal microscopy, one photon excitation can excite fluorophores above and below the focal plane, as depicted in this bottom left picture, and then filters out the non-needed fluorescence. However, in two-photon excitation, the excitation occurs almost only at the focal plane due to the rapid fall-off in excitation probability outside this point. This emitted fluorescence resulting from the excited state is then collected to form an image as shown in the bottom right image. Finally, the laser scans the specimen and repeats this process point by point to build a 3D image with high spatial and depth selectivity. So TPE also provides a 3D reconstruction, allowing clear discerning of structures up to 1 mm and at different imaging depths. It only elicits fluorescence at the focal point due to its nonlinear nature. So now we're going to go through TPE's advantages over confocal microscopy, starting with the key difference between both techniques, which is that confocal microscopy excites throughout the specimen, but then collects only at focal plane using the pinhole, as opposed to TPE, which excites fluorescence only at the focal plane organically due to its nonlinear nature. This translates to a couple of advantages. First, the fact that we are using longer wavelength photons would result in deeper imaging, because infrared light is able to penetrate deeper with less scattering. Also, focused infrared pulses provide precise imaging only where needed, thanks to its nonlinearity, which would mean better spatial selectivity. This allows us not only to get rid of the pinhole, but also reduce photo damage that is caused by exciting parts above and below the focal plane. And the fact that we don't have any excess light to filter out with the pinhole, we actually can now move the detector itself forward into the light path of the microscope and closer to the specimen, which improves the collection efficiency of scattered fluorescence. So to summarize, TPE offers many advantages over confocal microscopy due to the excitation being limited mostly to the focal area, which means less scattering and bleaching, and near-infrared light penetrating deeper into the tissue. So given these advantages, one might ask why wouldn't we always use TPE over confocal microscopy? Well, it also comes with its downsides, namely the increased equipment complexity, for example. It requires high power, ultra-short pulse lasers and sophisticated detection systems, adding complexity and cost. Besides that, although it practically eliminates photobleaching outside the focal plane, its intensity can still cause significant photobleaching at the point of focus, since high photon density is necessary for it to work. Other than that, not all fluorophores are optimal for two-photon excitation. Their efficiency can vary widely, necessitating empirical determination of suitable excitation wavelengths. So it's best suited for specific applications requiring deep tissue imaging or minimal photo damage, reducing phototoxic effects, rather than a universal solution for all types of fluorescence microscopy. Finally, TPE does not improve spatial resolution over a well-aligned confocal microscope, and may cause greater photo damage in thin samples with visible fluorophores. So TPE's advantages over confocal microscopy apply mainly to thick samples, ranging from around 100 to 1000 micrometers, such as brain slices or whole organs, or in vivo imaging such as neural activity in the brains of live animals due to its deeper tissue penetration and reduced photo damage. So for this slide, we're going to go through the specialized applications that we talked about earlier. Though confocal microscopy does a great job with samples up to 100 micrometers, as we see in this picture of an 8 micrometer section of a mouse optic nerve, where multiple choroidites are clearly depicted in high resolution, it wouldn't be able to do that with thicker samples. And that's where TPE comes into play, allowing deep tissue imaging up until 500 to 1000 micrometers for imaging neuronal tissue, immune cell dynamics, kidney function, cancer biology, and developmental biology, among other applications, giving clearly better results than confocal for thicker samples, as we see in this image of a 70 micrometer specimen. TPE is also great for in vivo studies, thanks to its low phototoxicity. It could be used in longitudinal studies of neuronal activity, immune cell dynamics, and metabolic processes, as well as investigations into disease progression and therapeutic responses in living organisms. It also enables precise optically based simulation experiments, such as the activation of cage compounds for spatially controlled release of neurotransmitters or other signaling molecules, allowing sophisticated investigations into neural circuitry and behavior through techniques like optogenetics. So to summarize, TPE enables deep tissue imaging with reduced phototoxicity and precise localized photoactivation, facilitating advanced studies in various biological processes and neural circuits. To conclude, the authors have highlighted several key advantages of two-photon microscopy. This technique offers highly localized excitation, which minimizes photo damage and allows for precise photochemical applications. It's particularly effective for dynamic imaging of live cells, intact tissues, and small animals, excelling in deep tissue imaging due to reduced light scattering. Moreover, existing confocal microscope can be adapted for TPE with the addition of non-D-scan detectors and external pulsed IR laser sources. However, it's important to note that two-photon microscopy does not improve spatial resolution compared to well-aligned confocal microscope, and may cause more photo damage in thin samples with visible fluorophores. Looking ahead, ongoing advancements in laser technology, detector improvements, and in vivo imaging will very likely broaden the applications of two-photon microscopy. That was it for our presentation. Thank you very much for listening, and feel free to ask us any questions.